I am Matt Pillar. I'm happy to welcome you to the first of our 24 series of Bioprocess Online live digital events. It's a long and expensive journey from molecular discovery to the clinic, making it all the more important that cash and timeline conscious biotechs, and aren't we all, take steps to expedite the discovery and selection of the most promising molecules for development. That's our focus today. And we've got a couple of great guests lined up for this discussion on R&D process efficiency. I know they're great because they've both been guests of mine on the Business of Biotech podcast. For the past 2.5 years, Nadim Ahmed has been president and CEO at Cullinan Oncology, a clinical stage company with a diverse array of large and small molecule candidates, addressing an equally diverse array of cancers. When I first talked with Nadim back in, I believe it was 2021, he took the company over with, I think, 1 clinical program, quickly grew it to 3, and now the company has got 7, I believe, candidates in the clinic. Before joining that R&D intensive endeavor at Cullinan, Nadim spent 11 years leading hematology and oncology programs at Celgene and BMS, and a whopping 18 years with GSK prior to that. Apparently, he started his career when he was 9 years old, or somehow he's avoided aging like a president, as this industry often does to us. Dr. Robert Stoffel was VP of Research at Horizon Therapeutics when it was finally acquired by Amgen last quarter. That's a title he retains at Amgen, the big bio juggernaut that needs no introduction. Similarly to Nadim, Bob's youthful appearance deceives the nearly 18 years he spent as director at AbbVie, his 7 years at BMS prior to that, and the 3 he spent doing postdoc research at Duke after earning his PhD in chemistry at Penn State. Nadim and Bob have graciously agreed to subject themselves to our questions for the next hour or so, and I can't overstate that your questions are more important than mine. So please don't hesitate to ask them earlier and often. Bob and Nadim, welcome. Thank you so much for joining us.
Great to be here, Matt.
Yep, thanks for having us.
It's my pleasure, and I appreciate your time. I want to start at a high level, and then we're going to drill down. And Nadim, since you're big on my screen, I'm going to start with you. I'm going to direct this question to you. If you could, I mean, Cullinan, you and I have talked in the past, and I know that Cullinan is exemplary of, as I said, a very research-intensive biotech. So if you could start by just sort of describing for us in real terms what the discovery process looks like at Cullinan.
Sure. I would argue that our process is quite unique, and I would say split into 2 areas, actually. So one is around target identification, and then the other one is around how we source innovation internally or externally. So starting with target identification, our discovery process really focuses on identifying what we think are high-impact cancer targets. And once we've found those targets, we then think about how do we address those targets. So for us, it's always target first, modality second, rather than the other way around, which is quite different to many of our peers. And we also focus on molecules that activate the immune system or target key oncogenic drivers. And our other go, no-go gate is pursuing molecules that have the potential, at least, to either be first in class or best in class. And so that's really important for us. And as part of that, our discovery teams really front load as much experimentation as possible early into the process across the R&D continuum. And then we further inform our efforts there through our translational research work, which we start very early on in the process so that we can get very early and deep understanding of mechanism of action, potential toxicity, dosing parameters, PK, so that this way we're only advancing the most promising molecules into the clinic. And so this kind of rigorous early experimentation, we would argue actually allows us to increase our probability of success as we go from discovery right through to development. The other aspect I spoke about was how we source innovation. So we call it innovation without borders, which really means we're very agnostic to the source of innovation. I mean, my direction to the team is get me the best innovation, whether it's internal or external, it doesn't matter. And I think our teams are very good at doing that. And so we've really embedded external collaboration into our research model. as an example, so that we can continue to advance ideas that we think will really benefit patients. So we have our own internal discovery efforts, but we also have exceptional engagement with industry and academic partners, so that together we can use the full ecosystem to deliver what we think are the most promising cancer therapies. And so, I've described our process, and then maybe I'll just give you a proof point, and you alluded to it. So I would say our process has yielded significant results. So we've gone from one program in the clinic about 18 months ago to 6 programs by the end of 2023. And it's a very highly diversified pipeline, a range of targets, a range of modalities across hematologic and solid tumors. And so that gives us multiple shots on goal, but not just multiple shots on goal. I would argue multiple high quality shots on goal, because of all the rigorous go, no go criteria that each of those molecules and programs had to pass before they even got into the clinic.
Fantastic. Real quick follow-up on that. You talked about your internal research capacities, and then the focus on looking for opportunities externally. Are those separate teams, or are they the same people?
No, it's the same people, which is great, because then you don't have often in the larger companies, you have that kind of tension between, internal versus external. So the same teams and the same scientists, work on our internal discovery efforts and also our external efforts. And that really was very deliberate, because it means that it really makes the team very agnostic to the source of innovation. And they really follow this model of let's get the best innovation wherever it comes from.
Yeah. Bob, I want to flip that question over to you. That first question to sort of describe the process. And I know I feel almost bad asking you the question, because you're in a unique position, having Horizon having just been acquired by Amgen. So maybe that's maybe it's in flux. I don't know. But you've got plenty of experience in the arena, both at Horizon and prior. So I wonder how you might tackle that one.
Yeah, I mean, so they're very similar in this regard, which is really you start with the patient. So it's always patient first, and that's what generates the disease states that you want to think about, as well as the target identification. So within Amgen, they've invested for a very long time in the genetics of disease, and they have a very large human database for analyzing this and really understanding on the genomic level, what pathways and targets are involved in the disease state. So from there's also been a very significant investment within the company in terms of like artificial intelligence and machine learning. So really trying to get towards the best therapeutic. And just as we just talked about with Nadim, really being agnostic to the modality. So really trying to figure out, okay, what's the best way to get to the disease state in the patient with this target? So there's a lot of effort in terms of, say, small molecule, trying to generate drug-like properties, as well as an efficacious molecule. And then even more so over the past few years on the biologic side, really using an iterative process of design, like artificial intelligence design, machine learning, and then testing in the lab to really get to the best therapeutic to advance into human testing.